All right. Good morning, biologists. Well, it's morning here. It's first hour. I'm in the lab. No Roxy. Here is the screencast to accompany the 9.4 reading guide. I'm going to just briefly touch on the cloning part and a little bit about the recombinant DNA, but you should rely on your textbook to get mainly that first page of information, and there's also some things that you're supposed to kind of think about and just kind of put down your own thoughts, so make sure you do that part. The main thing I want to go through is the steps of the genetic engineering process, which is on the back or the second page. So first part talks about cloning. Entire organisms can be cloned. Actually, the very first cloning of a mammal took place in 1996, and that was a sheep, and that sheep's name was Dolly. And make sure you go through the steps in the cloning process. That is right from your textbook. We'll move on to our next slide. As you are writing down the steps, the 5 steps of the cloning process, you can refer to this diagram to kind of put a picture with those steps. And then something to think about, and make sure you jot down your answer. How would a clone of you be similar, and how would a clone of you be different? All right, definitely need to know this term, recombinant DNA, is DNA from more than one organism. And I always remember it's a recombination. The word recombinant comes from recombination. It's a new combination of DNA. Here, what the picture is showing is you have a mouse. Oops, I gotta get my pencil, sorry. Here is your mouse. Here is a glowing jellyfish. And what we are able to do is take that glowing jellyfish gene from this organism and insert it into the mouse, and voila, we have glowing mice. And we are going to be doing this lab not with mice, with E. coli bacteria. It's called the P-Glow lab. So basically we're going to have, instead of a mouse, we're going to have bacteria. We are going to insert the glowing gene, and we are going to make our bacteria glow in the dark green. All right? All right, then on the back, the second page of notes, it goes through the steps of the genetic engineering process. You need to know these steps in order. First is to cut the DNA using restriction enzymes, of course, because we know restriction enzymes cut DNA at specific sequences. We make the recombinant DNA, so that's the DNA from 2 or more sources. We clone it, which is basically just letting it reproduce. And then we screen the cells. And we screen the cells using antibiotics. And you need to understand why we do this. And then the outcomes if we have antibiotics present, if we don't have antibiotics present. But I'll go through that, all right? So those are your steps. I'm going to go through each one of these, but I did take a screenshot of my notes with the pictures that I drew, and I would suggest pausing this at some point, getting this down. You can either come back to this, or I did put it at the end of the screencast as well if you want to kind of get the word part of the notes down as you go through each slide and then do the pictures later. I also included what color flares I used to make this diagram possible. Haha. All right, so the first thing that we do is cut the DNA. So we choose a restriction enzyme. It needs to be the exact same restriction enzyme that's going to cut out your gene of interest, and the same restriction enzyme then is going to open up the plasmid. I feel like I missed a slide talking about plasmids. Hold on one second. I want to make sure I discuss plasmids. Let me go get that slide and I'll talk about it next. I don't know where the slide went, but I'm just going to draw it right here. A plasmid basically is a naturally occurring piece of DNA that's found in bacteria. So this is our bacteria cell. And this is the picture that is at the very upper right-hand corner of this page in your notes. It says draw a bacterial chromosome and plasmids inside the bacteria below. So what I would draw, what I drew, the bacteria have a main chromosome, which is right there, but then they also have these extra little circular loops of DNA that are called plasmids. And there can be numerous plasmids in a bacteria cell. And these plasmids are perfect for being able to use as a vector. A vector is a generic term of anything that is used to carry DNA from one source to another source. Viruses can be vectors. There's lots of things that can function as vectors. We're going to talk about plasmids as being our vectors. So basically, you take your gene of interest. Let's say it's that glowing gene from the jellyfish. So here is some, the strand of jellyfish DNA, and this is the part that has the sequence for the glowing. You use a restriction enzyme to cut that out. You use the same restriction enzyme to open up your plasmid, and then you insert your gene of interest into the plasmid. So on the picture, I had drawn where it cuts, and then actually this is step 1 and 2. Inserting it is showing the plasmid in pink with the gene of interest, that extra recombinant DNA with our gene of interest inside the plasmid then. All right, so after we've cut it, using our restriction enzymes, we have our sticky ends exposed, and because you're using the same restriction enzyme, they're going to be complementary, and next week we're going to do an activity where you're going to actually be doing this with paper so you can see the complementary sticky ends. The genes then are added, the complementary ends stick and are bonded together, resulting in a recombinant DNA. All right, step 3 is to clone the gene. Literally, this just means to allow the bacteria to reproduce. Remember, bacteria reproduce asexually, and it occurs very, very quickly, so you're making millions of copies of this gene. So this is our original bacteria. It has its normal bacterial chromosome. It has the plasmid with our gene of interest, and you'll notice there's another little section that's in there. I'll talk about that in the next slide. But cloning just means that it reproduces. You get 2, you get 4, you get millions and millions and millions of copies of your gene of interest. Now, this last step is, it turns out we call it the last step, but it's done during the reproduction cloning stage as well. You basically also add not only your gene of interest to the plasmid, but you also add a resistance to an antibiotic. And when you grow the bacteria, remember you're growing bacteria. I think you guys in middle school at some point have used Petri dishes and the agar and plated bacteria, or gone around the school and gotten different bacteria samples. You're growing it on that agar. Normally it's just food, but what happens in this screening process is in some of the plates we add antibiotics to it. And if an antibiotic, you got to think of antibiotics, okay? Antibiotics are prescribed when you have a bacterial infection to kill bacteria, all right? So the idea is that we don't want to have all of the bacteria, because remember, or I don't know if I said this, but the plasmid is not taken up by every single bacteria cell. Sometimes the plasmid isn't taken up. We don't want to mess with the bacteria cells that didn't take up the plasmid, so we want to kill those off. So how we kill those off is we use them with the antibiotic, all right? So basically, you're screening or testing to determine if the recombination was successful. Was that vector or plasmid taken up by the cell, and is the gene of interest being expressed? So if we look at these different plates, obviously if we don't add antibiotic to the plate, you're going to get millions and millions and millions of bacteria, ones that took up the plasmid and ones that didn't take up the plasmid growing here because there's nothing to kill it. Here, you have, in both of these situations, you've applied antibiotics. If the vector was not taken up, that's what this one says, you are going to have no growth because there's nothing to make that. Remember, the other thing that we added to our plasmid was a resistance to antibiotic gene. Resistance to antibiotic gene means that it is going to not be able to grow or be able to grow on an antibiotic, all right? So if your bacteria didn't take up the plasmid, no growth here. Our last situation, this is the one that we're interested in, okay? Anything that grows on this plate, it has antibiotic. If the bacteria is growing, it means it took up the plasmid, it is expressing the gene of interest, and also causing it to be resistant to antibiotics because we added that. Normally it wouldn't be able to grow on an antibiotic, all right? But now all of a sudden it is. So that means that we were successful. The plasmid was taken up, and we can assume that all of these bacteria have our gene of interest. Now, the glowing jellyfish gene, they're also going to be glowing in the dark. We'll see that when we get to our lab. But you have to consider, and you might think, well, who cares if some bacteria grows if they didn't take up the plasmid? If you are a big drug company, let's say that you're producing insulin, this is how insulin is produced for people who are diabetic. You don't want to be messing with a bunch of bacteria that didn't take up the plasmid, that don't have that insulin gene. You just want to be concerned with the one that the ones that have the insulin gene. So that's why you use this process to ensure that anything that's growing on these plates has that insulin gene. All right? I think the last slide is one more picture of the notes with the drawings if you want to make sure that you get that. So again, the steps of the genetic engineering process. First, we're just cutting it using a restriction enzyme. You have to use the same restriction enzyme on your gene of interest and your plasmid. Then we are inserting or making the recombinant DNA. So we're inserting our gene of interest into our plasmid. Then we're allowing them to reproduce, so we're just getting tons and tons and tons of more bacteria. I should say, too, that we're taking this and we are putting it in another bacteria cell, okay? So we originally took it from bacteria cell, then we would insert this into a bacteria cell, allow it to reproduce, and then the third step, we have added the antibiotic to the growth medium, and we're concerned about this plate here. Those are the ones that have taken up the plasmid and are expressing whatever our gene of interest is. All right, so be ready to discuss this tomorrow in class, and you'll be working more with these topics next week when we do our E. coli insulin factory activity that is also going to be part of an assessment grade. So I'll talk more about that Friday and early next week. Thanks, guys.